Welcome to this lecture on the sleep epidemiology, the quest for causality. This presentation has been created with a teaching objective in mind, that is to use a real case study to describe the main challenge of observational epidemiology, that is to determine a cause-effect link between an exposure and an outcome. In this slide, we have some learning outcomes, and I won't spend time now because we will check them one by one at the end of the lecture. Research of the circadian rhythm and of sleep is mainly a post-war phenomenon. The studies of Freud and Jung represented until then what scientists studied and knew about sleep. This slide presents the exponential rise in published science in the field of sleep research, with milestones discoveries on its path. From the early description of rapid eye movement stage of sleep, to the first clinical description of obstructive sleep apnea syndrome, the critical discovery of the sleep-wake pacemaker in the suprachiasmatic nucleus of the hypothalamus, to the studies of the effects of excessive daytime sleepiness and fatigue, and its correction by using continuous positive airway pressure in those with OSA. More recently, the epidemiology of sleep has given insights into the population aspects of sleep disturbances, revealing the long-term consequences of sleep deprivation and the public health implications. In 2017, these three scientists, Hall, Rosbach, and Young, were awarded the Nobel Prize in Physiology and Medicine for their discoveries of molecular mechanisms controlling the circadian rhythm, bringing to the fore this new dimension of science, the study of human sleep. The Nobel Prize was just rubber stamping the exponential speed of development of the science of circadian rhythm and of sleep medicine. The case study I'm going to present to you illustrates the course we followed with our team over the past 15 years in trying to establish a causal link between short duration of sleep and risk factor for cardiometabolic diseases and other hard vital outcomes. As in most cases, the quest starts from a chance observation. As cardiovascular epidemiologists, we were well aware of the emerging epidemic of obesity in the Western world at the time, as indicated by a steady increase in the average body mass index of the American population over time, and the associated rise in the prevalence of obesity, as you will see in the top panel. As novices in the field of sleep medicine, we were tasked to develop a new research program on sleep. Clinical epidemiology was our field, so the first thing was to look at time trends in the average duration of sleep, starting from the US, where data was available, as in the bottom panel. We found that Americans had steadily reduced their average daily sleeping time almost in parallel with their average weight gain. Could anyone reasonably conclude that there was a causal connection between sleep reduction and weight gain? But the answer is definitely no. This is due to the flaw inherent to the correlation between an average exposure and an average outcome, which introduces the concept of ecological fallacy. This is an error in the interpretation of data that occurs when inferences about the nature of individuals are derived from inferences about the group to which those individuals belong. This slide presents ecological data relating average self-reported duration of sleep obtained by questionnaires on the x-axis and self-reported poor health, also by questionnaire, on the y-axis, in samples of college students taken from different countries using the same methodology. Although the results do not prove cause-effect, the data tend to suggest that there is a lower threshold for average duration of sleep, below which there is an increase in ill health. Whilst the temptation to infer some sort of causality between these two measures is there, when you sleep too little, then you experience more ill health. This set of data is not only open to the ecological fallacy, but is also cross-sectional. Exposure and outcome were measured at the same time point. We therefore decided to move away from ecological evidence and to examine data taken from individuals. The question we wanted to answer was, how much sleep do we need? Over the history, Humans have adopted different patterns of sleep. We know that if we do not sleep, we eventually die. However, how much is the optimal requirement? We don't really know. If we study current westernized populations, we detected a bell-shaped distribution of hours of sleep, with an average sleep duration between being clustered around 6 to 8 hours per night, but with an average of about 7 hours per night. And this is seen in the UK population of men and women on the left-hand side. We could also see about 8 hours per night if we only consider the duration of sleep at weekends, as seen in these American population samples. Whilst the majority of people do sleep around the average, 1 in 4 either sleep less, and we call it by convenience short sleepers, or they sleep more, and we call it long sleepers. Having established that sleep time can be measured in individuals, and many studies have somehow collected such data, we develop an a priori hypothesis. Does short duration contribute to the development of chronic disease? And in particular, cardiometabolic diseases. We developed our model where the exposure, short sleep, through a variety of pathophysiological mechanisms could lead to an increase in risk factors, leading to an increase in morbidity and mortality. This slide reports the results of a systematic review and meta-analysis of cross-sectional population studies in which both sleep duration, mostly self-reported, and prevalence of obesity had been measured in large studies of children and adolescents on the left and adults on the right. The graphs used are shown as forest plots. I shall use these a few times later, so for those who are not familiar, I'm taking some time to take you through the interpretation of these plots. They report for each study the average outcome measure, in this case odds ratios, and the correspondent 95% confident intervals of the association between the exposure, in this case sleep duration, and the outcome obesity. The likelihood of being a short sleeper if you're obese is measured by the odds ratio. Greater than 1 indicates greater risk. If the 95% confident intervals do not encompass 1, then the association is statistically significant, so is unlikely to be due to chance. The pooled effect at the bottom in bold is the result of a meta-analysis of all studies put together, allowing for the differences between larger and smaller studies. Both in children and adolescents on the left and in adults on the right, those with obesity were more likely to be short sleepers. The association was strong, as indicated by the strength of the odds ratio, 1.89 and 1.55, and unlikely to be due to chance, as the pooled 95% confident intervals were not including 1. The meta-analysis also showed that there was no heterogeneity, well, there was some heterogeneity between studies, but no publication bias. Can we say from these results with confidence that short sleep is causally associated with the development of obesity? The answer is no, since these are all cross-sectional studies in which both the exposure and outcome are measured at the same point in time, so we can't remove the risk that, dissect out the chicken from the presence of the egg. Observational studies are unable to answer one fundamental question: is sleep deprivation a cause, a consequence, or a symptom? In the study of sleep deprivation and obesity in children and adults, the real possibility of a bidirectional model of biological plausibility requires researchers to dissect out the direction of causality. From one side, it is possible that sustained sleep deprivation through a chain of events will lead to an increase in energy intake and then a reduction in energy expenditure, which in turn will be compatible with weight gain. On the other hand, if you start from the right-hand side, overweight and obesity are conditions often causing sleep disordered breathing, ensuing sleep fragmentation, and reduced sleeping time. This is often referred to as reverse causality. Therefore, it is important that observational studies on the effect of sleep duration only takes into account prospective studies in which the exposure of sleep deprivation precedes the development of weight gain or obesity, as well as other outcomes of interest. Given the density of the content, I think it's probably time to take a break. I think you can take as long as a break you like, and then restart the recording whenever you're ready. This large forest plot summarizes all the evidence of a prospective longitudinal association between sleep duration and incidence of obesity in population studies using children and adolescents. They were published up to mid-2018. It was a time when we did publish a study, in which sleep duration was measured at baseline in non-obese children, and the risk of developing obesity was recorded prospectively in the years to come. These studies unequivocally show the temporal sequence whereby short duration precedes the development of obesity. The exposure, short sleep is strong, 1.57, and the association is unlikely to be due to chance. So we can then say that these results are unlikely to be due to reverse causality and are compatible with the cause-effect relationship being strong, and also respecting the temporal sequence whereby the exposure precedes the outcome. This recent additional meta-analysis of a similar kind adds further criteria in favor of causality. The consistency of findings. This is a study showing that studies on short sleep in prospective design are predictors of obesity, not only, as we've seen before, in everyone, mostly westernized world, but also in cohorts of Chinese children and adolescents, indicating the consistency of the evidence across different ethnic groups and geographic location. This is a busy slide, and I'll take it slowly. These are some of the studies demonstrating the direct effect of sleep deprivation and poor quality of sleep on glucometabolic mechanisms. All use randomized crossover trial designs in normal volunteers, studied in strictly controlled laboratory conditions and subjected to short-term sleep deprivation. If we go in order, let's look at the first bullet point of the study. In the first example, 2 days of 4 hours per night sleep caused a reduction in leptin and an increase in ghrelin, hormones regulating hunger and satiety, with a net result of an increase in hunger and appetite. Furthermore, the effect was dose-dependent. In other words, the shorter the sleep, the greater the reduction in leptin secretion. In the second study, it shows an effect of short-term sleep deprivation on measures of glucose tolerance and insulin sensitivity. In all parameters studied, short-term sleep deprivation causes a reversible glucose intolerance and insulin resistance. In the third study, and this is a trial of 3 weeks of sleep restriction and concurrent circadian disruption in young volunteers, which caused an increase in postprandial glucose response that was due predominantly to the reduced pancreatic insulin secretion. The fourth study is the first molecular evidence in a randomized trial of a plausible molecular mechanism showing in vivo in these volunteers that the reduced insulin sensitivity seen during short-term sleep deprivation is due to an impaired phosphorylation of PAKT with reduced insulin receptor transduction and therefore insulin resistance. And finally, 1 week of 5 hours per sleep versus 8 hours causes up and down regulation of the expression of a variety of genes associated with circadian rhythm, sleep homeostasis, but also in addition with a gene expressing oxidative stress and metabolism, indicating a reversible effect of sleep deprivation on the gene expression involved not only in the regulation of the sleep-wake cycle, but also in cardiometabolic functions. So we have plausible mechanisms, dose response, and reversibility of the effect. The effects are also seen on behaviors. Participants reporting later sleeping time were more likely to consume sugary caffeinated beverages and high energy dense, nutrient poor foods, and to consume more food throughout the day and into the nighttime hours compared with their early sleep timing peers. And this adds a plausible mechanism of behavioral inference. So this slide indicates the progress toward the steps of recapping the line of evidence for causality we followed between sleep deprivation and disturbances that might cause metabolic consequences. We've shown that sleep deprivation causes energy, reduction in energy expenditure and an increase in hunger and appetite, by that supporting the view of a plausible mechanism leading to obesity. The obesity is a condition of insulin resistance and glucose tolerance, as we have shown to be caused by sleep deprivation. If that is sustained over a longer period, it is likely that that may lead to development of type 2 diabetes. And that was our next step, to try to demonstrate the potential link between sleep deprivation and type 2 diabetes. At this point, we follow our a priori hypothesis. Having done the studies before, we only concentrated on the prospective evidence that sleep deprivation may predict the incidence of new cases of type 2 diabetes. And this is a meta-analysis indicating not only the effect of duration of sleep, but also quality of sleep, like difficulty in initiating sleep and difficulty in maintaining sleep, on the incidence of new cases of type 2 diabetes. For all these three exposures of sleep, the shorter the sleep or the poorer is the sleep, the greater is the chance of developing type 2 diabetes over a period of follow-up. So the evidence is strong and unlikely to be due to chance. As in metabolic, also in other conditions of the cardiovascular system, sleep has an effect. Here, sleeping less than the norm leads to the development of high blood pressure, as shown in this meta-analysis of prospective studies, with a risk of 20% higher in those regularly sleeping no more than 5 hours per night. Short duration of sleep has an effect on blood pressure very early in life, as well as indicated by the study in adolescents. They were followed for 5 years in a cardiac study, where short duration of sleep measured, this time objectively with actigraphy, predicted the rise in blood pressure occurring with age and improved sleep maintenance. Furthermore, if we see the other side of the spectrum of the life course, in a study of older men who underwent full polysomnography, so with the ability to stage sleep, they were then followed up for 3.5 years. Short sleep was shown to be a good predictor of hypertension, as shown in the meta-analysis. However, the additional information, the study, that they were able to show that the cause of the shortened sleep duration was due to reduction in the slow wave sleep stage, which is considered to be the most restorative and deeper stage of sleep. So time sequence strength added consistency. This is a randomized clinical trial now, and one of the very early, a few available control trials of sleep extension in healthy volunteers. This time it's trying to extend the duration of sleep as an intervention. And 2 groups were randomized to either maintain their sleep duration to about 6.6 hours per night, or extend it by just 1 hour per night. 24-hour blood pressure was measured at baseline and after 6 weeks of intervention. This slide shows systolic and diastolic blood pressure baseline in blue and follow-up in red, in the sleep maintenance group on the left and the sleep extension group on the right. And the period of sleep extension caused a significant fall in blood pressure across day as well as across night. The intervention extended total sleeping time by an average of 30 minutes per night. A randomized clinical trial is the ultimate proof of causality. Although it's a short term, shows a reversible effect to the removal of the exposure. We have so far seen a quick summary of evidence to support the view that short duration sleep causes a variety of effects, from metabolic, vascular, hormonal, molecular, leading to the development of numerous risk factors, obesity, hypertension, diabetes. It is therefore reasonable to postulate, and we follow our a priori hypothesis, that long-term sleep deprivation might be found to be associated with a higher risk of cardiovascular outcomes. This slide reports the summary of the epidemiological evidence in prospective studies showing that sustained sleep deprivation leads to an increased morbidity and mortality from cardiovascular causes. We've seen, and this is all based on large meta-analyses, including a large variety of patients there. We have seen already that sleep deprivation predicts the incidence of obesity, hypertension, and type 2 diabetes. In additional meta-analysis, we also reported an increased risk of developing fatal and non-fatal coronary heart disease and stroke, and surprisingly premature death within 12 years of observation. And these effects are strong and significant from a public health viewpoint. For the latter evidence, the effect on mortality, there was debate when we published the study on whether a single assessment of sleep duration by self-reporting would be a tool reliable enough to imply that the sleep deprivation was sustained during the follow-up period. In fact, sleep duration is one of the behavioral characteristics that can change substantially through the life course. We therefore decided to look at the data in a different way in the Whitehall 2 study, a cohort of British civil servants representative of the British adult population. The study repeated observations every 5 years so that we were able to measure the exposure sleep duration at 2 points 5 years apart and to classify individuals in groups according to their sleep duration. The reference group is made of individuals who reported the same habitual duration of sleep on 2 separate occasions. Those who reduced their sleep duration from 6, 7, and 8 hours to less were considered sleep deprived. Those who increased from 5 to 6 hours to more, defined as improvers, and those who had increased from 7 to 8 hours to more, labeled as long sleepers. All were then followed up for 8 years, and the vital outcomes were measured. Now, irrespective of the model used to adjust for confounders, both those who reduced their sleep over time and those who increased their sleep over time increased the risk of death from all causes. However, those who reduced their sleep over time died mainly of cardiovascular disease, whereas the excess mortality in long sleepers was attributable to non-cardiovascular causes, suggesting some specificity of effect. This scheme summarizes the wealth of evidence accumulated in the past 20 years, extending the concept that sleep disturbances are not only a problem for the brain and confined to the nervous system. Sleep disturbances involve almost every system and organ of the body, with far-reaching consequences in the short and longer term. And on this graph, I expect you to take another break, take it as long as you like, and we go to the last part of the talk in due course. The third part of this talk is addressing now the summary of how to tackle the concept of causality in observational studies. To find an association is not necessarily defining a causation. Association may be present in the absence of a true cause-effect relationship. Epidemiological study designs, systematic random variations, and alternative explanation may support the false belief that an association is causal. So there are different ways of addressing the problem. First of all, let's see what models challenge the concept of causality. We can find 3 situations. We can find a situation in which the relation between exposure and outcome may be confounded by a third factor, which is related both to the exposure and independently to the outcome. We can find also the confounding effect created by a mediator, they may blur the association with two different factors. And finally, there could be a confounding by exposure, which is related independently to two factors, and the two factors seems to be associated with each other. Let's give you three examples. The first example, I use the example in the past that low social class and low level of occupation was usually more likely to be associated with the incidence of lung cancer. And for years now, there's been thinking, why is this the case? And eventually, the clarification is that there's no particular plausible mechanism whereby occupation will lead to lung cancer independently, but there was a confounding effect of smoking, in that smoking is associated with the occupation. Low level of occupations are more likely to be smoker or were smoker in those days. But independent of that, smoking causes lung cancer. And therefore, if you don't measure the variable smoking, you might be misled by thinking that occupation is a causative factor for lung cancer. The other classic situation is a diet causing coronary heart disease. If you take any diet, not all cause coronary heart disease, but these effects of certain diets are mediated by factors like animal fat and a rise in serum cholesterol. And in reality, is the serum cholesterol the causative factor, CHD, which is produced by specific diets high in animal fat. And finally, the fact that we may find an association, and we do find an association between alcohol intake and lung cancer very often, and that has no plausible mechanism to support it unless you are aware that alcohol intake is associated with smoking as a behavioral factor, and that smoking is a cause for lung cancer. So to interpret observational epidemiology, we need to follow first the rule of ruling out. The first three things to rule out are the rule of chance, in other words, that statistical inference will assess whether it is by chance that you find an association. And there are different methodologies. We're not going to go into detail. So rule out the rule of chance. Rule out also the rule of bias. Bias is to get an incorrect information, either due to the selection of people in your groups or the wrong information gathered in terms of exposure or outcome. And in the sleep epidemiology, there's been a lot of discussions about the recall bias, differential recall bias, as in early studies, most of the exposure or duration of sleep was reported by questionnaire, with the risk you might not report very well your average sleeping time. And finally, to rule out the role of confounding, as we've seen earlier on, either by by adjusting for known factors that might explain the spurious associations with age, sex, ethnicity are the ones that are very obvious, or possible factors like socioeconomic status or deprivation, remember occupational lung cancer and smoking, or by unknown factors that you're unable to measure, like the genetic factors. Well, having said that, in the 50s and 60s, Sir Austin Bradford Hill came to organize some criteria in a logical way to address the quest for causality. And these are known as the Bradford Hill criteria for causality. Sir Austin Bradford Hill was an English epidemiologist and statistician who then pioneered the randomized clinical trial methodology. But in those days, there wasn't still quite a lot of evidence through randomized trials. And eventually was mentored to Sir Richard Dole, and together they eventually proposed the demonstration of the causal connection between cigarette smoking and lung cancer without ever doing a randomized clinical trial. So the Bradford Hill criteria are a set of criteria that tend to evaluate the strength of the evidence in favor of a cause-effect relationship in an association. This table lists them all and then you will keep it and read it in your own time, but basically are a series of qualities: strength, temporality of the association, dose response, reversibility, and so on, which are the red banners I've used on my slides as I went along in the case study. And there is no particular rule on how many you need to meet. The more you meet, the closer you meet them, the more likely you are that you're inferring causality from your association. And you have to go through this process. So in summary, I want to say that using our case study, the association between short duration of sleep and cardiometabolic risk factors and outcomes may reflect causality, as the effects are strong with large relative risks. They are consistent, in other words, they are confirmed in different populations for several endpoints. They show a temporal sequence, short sleep preceding the endpoints. And they show dose response, which is consistent very often in sleep with a threshold effect. And there are biological plausibilities tested at several levels with potential mechanisms in the genetic, molecular, cellular, physiological, and metabolic areas. And there is some evidence of reversibility confirmed when tested in controlled clinical trial conditions, at least in the short term, as we've seen for insulin resistance, glucose intolerance, and hypertension. So going back to the learning outcome, let's see what you should be able to do at the end of this lecture. You should be able to understand the limitation of observational research in establishing a causal link between the behavioral exposure and health outcomes. You will be able to understand the criteria that help imply causality in epidemiological research. You will be able to contrast the relevance of different study designs to imply causality. You will critically appraise published epidemiological research with a focus on sleep epidemiology. This is a general critical skill that everyone should have in reading a scientific paper. My personal suggestion would be to read methods and results and use your critical skills to assess what the conclusions of the study are. And then finally, define the criteria for causality when designing and interpreting observational epidemiology between an exposure and different types of health outcomes. I thank you for listening to me. I'm available on email for any clarification you might have throughout your perusal of this lecture.